pepsin active. Okay? And for proteolytic activation, for example, it's mentioned that for covalence modification, what happened? What is the source of this phosphor? Huh? No, the source of the phosphor. In covalence modification, you require energy. Okay? But here, for example, conversion of trypsinogen to trypsin, there is no need to energy. Okay? And another difference. This is important for me. This is a comparison between proteolytic cleavage and covalence modification. So, for covalence modification, you require energy. Here, no energy. Another point. For covalence modification, this is a reversible reaction. You can add a phosphor, take a phosphor. Kinase, phosphatase. Do you understand? But, for example, I'm trypsinogen. If you take some amino acid from me to become me trypsin, I cannot turn back to the trypsinogen. This is irreversible. Okay? You cannot, never, you cannot convert a pepsin to pepsinogen. Because when you have a pepsinogen, you take some amino acid from enzyme to become it pepsin. You cannot turn back. So this is an irreversible reaction.
Like infants and adults?
What? Infant, adults, what?
Baby and adult, like cell.
Yes, yes. It's an example, actually. So there is no actually way to turn back to the zymogen. Okay, so it means that this is irreversible. This is an example. What is important here? At first, site of synthesis is important. You should know that pepsin present in which organ? Stomach. The remaining are pancreas. Maybe from this table, maybe I ask you this question. All of the following molecules are active except whenever you have a gene and whenever you have a pro, yani inactive. Okay? Trypsinogen, inactive. Chymotrypsinogen, inactive. Trypsinogen, inactive. Procarboxypeptidase, inactive. Okay? But all of them considered as an active molecule. And about the site, faqat pepsin stomach. Other are pancreas. About the actually activator, I mentioned that this is a slide for actually, inshallah, in a metabolism of proteins, I will talk. Just know that because most of the time I ask the activation and inactivation, we talk about it in this slide. Okay? The active, inactive, and active is important. What about this slide? What is important? As I mentioned, the pink box are inactive. Pro-lipase, pro-carboxypeptidase, chymotrypsinogen, pro-elastase, trypsinogen. By this way you can find the inactive. But if I ask you, and you can find actually the answer of my question, all of these enzymes considered as an enzyme for digestion of protein. Okay, please tell me, as I mentioned, for digestion of protein, at first our body produced the inactive and then became activated. We have one general activator, can activate all of these inactive enzyme to active. Inactive to active. What is the name of this enzyme? Activate all of the digestive enzyme. I put in this slide. Entropeptidase at the beginning. Excellent. As you can see here, this is the key points. So if in your final exam, I ask you about the general activator of all of the inactive enzyme or zymogens, this is a trypsin. Is it clear? This is another question from this slide. And about the plasma enzyme, just I want to have a actually fast review, just 5 minutes. As we talk about this point, some enzymes, especially isoenzyme, are present inside of the cells. Okay. For example, maybe they are liver cell, bone cell, muscle cell. Also, you have a bloodstream. Please tell me when we talk about this one, because I want to repeat my question. As I mentioned, sometimes you have a non-functional plasma enzyme. At first, I should describe what is the non-functional. Non-functional plasma enzyme, yani present in plasma in a low concentration. Why? Because they don't have any role in the plasma. They have a role in a bone, in a muscle, in a liver, in a pancreas. Okay? When you observe this enzyme in the blood, these are non-functional. When you can see this enzyme in the blood, when you have a damage here, am I right? For example, this is the bone enzyme cell. Enzyme actually specific, bone specific enzyme. You have bone specific enzyme. After car accident, you have a fracture in your bone. So your bone became defective and they release the enzyme into the blood. So you understand, there is some damage to the bone.
Yes.
For example, again, in a car accident, not only your bone are damaged, but also your muscle may be damaged. It can cause a damage to the muscle cell. It can release some enzyme that's present in a muscle. Okay? This is the concept of non-functional. Functional enzyme, we didn't work with functional, also as a physician, because they have a function, like a blood clotting enzyme. But most of the, for example, liver disease, bone disease, pancreas disease, okay? They consider as a actually non-functional excellent, and as a physician, you should order the plasma. For example, as a physician again, you are suspected to the cancer of the pancreas. Okay. One of the marker you should order to the laboratory, you should ask, for example, pancreas is full of lipase, amylase, okay, or other digestive enzyme. As a physician, if you order, for example, the lipase concentration in a blood, when we say order, all the time you cannot take from bone, muscle, all the time you have a blood serum, okay? Then the pancreas enzyme of the serum is abnormally very high. What happened?
Sign of the
Damage to the pancreas. Maybe cancer. I don't know. Maybe any marker can, any actual molecule can damage the cell membrane of the pancreas cell. They can release some molecule. And for example, another question, it's better to continue with this is fine. We talk about this point. This is for more information. These are inshallah. For the next semester, we talk about this one. For example, ethanol can damage liver. Again, these are for more information because inshallah, for the next semester, you have one session. Only 2 and 3 slides, okay? Because when you whisper, I actually cannot contract, okay? So, for example, yes. Yes, also.
You can find this in all the body blood or
This sentence, sorry. excuse me, wait.
More
More information, again this one and this one. But I want to just describe, for example, why we say that, for example, in Islam, we believe that alcohol is haram, okay? Because ethanol can damage, your liver cell, and inshallah I will describe AST and gamma glutamate transferase, they present in a liver, okay? When they release into the bloodstream, yani your ethanol causes damage to the liver. For example, if you ask from your patient some drugs, they take some drugs, for example, phenytoins and other drugs, because these drugs can cause a damage to the liver, it can cause an increase of liver enzyme. And for example, you have actually increase in some enzyme of the bone, osteoblast. This is alkaline phosphatase. It means that you have a hyperactivity of your cells. And again, for example, if you have exercise, you have a high activity of muscle enzyme. I will talk about this point. And sometimes, sometimes some enzyme can excrete into the urine very easily, like amylase, okay? And as a physician, you see that, amylase is a pancreas enzyme. Maybe there is a problem in the pancreas. What you evaluate, for example, with the sonography, you say that, okay, pancreas is okay. So maybe there is a problem in other parts of the body. For example, normally, we excrete the amylase in the urine. If you cannot excrete into urine, what happens? It accumulates a lot. Okay? So maybe amylase is accumulated is as a result of renal failure. It's not as a result of pancreas failure. Okay? Inshallah, I will describe by details. And more information again. Just this slide. Okay? فقط this slide. I want to. This is my last slide actually. Just know that because I require some background information to understand the actually serum enzyme, I put it intentionally to become a little familiar. And inshallah, during one session, I describe the details. Just know that as a physician, I want to mention one example of serum enzyme or non-functional. For example, you have one patient who suffers from chest pain. Okay, very severe chest pain. Okay, maybe you are suspected to the MI. Okay, maybe you're suspected to the MI, myocardial infarction. How you can evaluate the myocardial infarction? You have some factors, some markers. Okay. One of the markers is creatine kinase. Creatine kinase is a muscle-specific enzyme, not only in skeletal muscle, in a heart muscle. But we cannot take a nose if, as a physician, you read the results. Creatine kinase is high. Absolutely, MI happen. Well, actually, it's maybe as a result of some damage to the skeletal muscle. You cannot take a nose. The source of creatine kinase is from heart muscle. Okay? So it's better to choose another marker that you can take an oath. Absolutely, after observation of this molecule in the blood, MI happen. What is this gold standard and marker? This is a troponin molecule. This is one regulatory molecule for controlling the muscle contraction. You can take an oath for this biochemical marker because C means that cardiac. Cardiac troponin 2 considered as the gold standard for the occurrence of MI. Why? Because fat in a heart is present, not in a skeletal muscle. Whenever troponin in the bloodstream is high, يعني absolutely you have a damage to the heart muscle. Okay? So this is the gold standard for the evaluation of the MI. Many thanks. Actually, that was the last session we had. We actually had a class together. And please, I don't know what do you think? Okay, please say prayer to have a peace in Iran, inshallah. Many thanks.
سمين يريد سمين يريد بمخه